week 7, or whatever week you may be doing this specific presentation tutorial. But today we're going to be discussing the respiratory system in somewhat a little bit of depth. So, if you've done human biology in a previous, unit or at high school, you're going to find the content of the respiratory system very basic and very easy. And that's not to say the respiratory system is easy or is basic. There are actually a lot of components to the respiratory system we're actually not going to cover today. But once again, it may be of interest to you, and you can do further research down the line in that specific topic. Now, guys, in the lecture you should have described a few things or went through a few things, and one of them may be and gained an understanding of the functional components of the respiratory system. You should have described the mechanics of gas exchange in and out of the lungs. Sometimes it's referred to as gaseous exchange. You should have described also the physical factors influencing pulmonary ventilation, and explained and compared various lung volumes and capacities. Now, guys, I'm going to show you a video today, 5 minutes long. It's going to cover a lot of the content in terms of not gaseous exchange, ventilatory respiration, and so on and so forth, and pulmonary ventilation, pulmonary respiration. So essentially that makes my job easier, and it's going to make a little bit more sense to you, as she can use props, drawings there and then on the screen. So it's actually a really good video for you to understand breathing, vital capacity, total volume, and all that type of stuff. So let's first go through and start talking about pulmonary ventilation. Well, pulmonary ventilation is moving air into and out of the lungs. As the name suggests, it's also referred to as inspiration and expiration. So step 2 is the external respiration. So gaseous exchange between the lungs, the alveoli, and the blood is referred to as external respiration. Okay, so that's when oxygen diffuses into the blood and carbon dioxide is in the blood diffuses into the air. Internal respiration is a little bit different. It's when the gas itself is exchanged between the systemic blood vessels and the tissues. And we refer this as oxygen into the tissues for ATP production and carbon dioxide in tissues diffuse into the blood. So it's essentially what happens at the cellular level. So a little bit of anatomy of the respiratory system. Well, we have the larynx and the trachea, so we know about that, or we should, from our lecture. We know about the bronchus and bronchi and the split of the bronchus. We then obviously know that the bronchus then branches into the bronchioles, and following the bronchioles, we have the alveoli. So we can actually see this process, the branching of the bronchial tree. So we have the trachea to the primary bronchi, to the secondary bronchi, the tertiary bronchi, the bronchioles, and the terminal bronchioles. So obviously it's important to know all those steps there. But if you forget, obviously it's just trachea, bronchi, and then bronchioles. So a little bit of anatomy of lungs. We're not going to spend too much time here, but this is essentially the inside and what actually consists of the lungs. Now you may or may not have gone through this in your lecture, but it's a good thing to kind of just rehash and look over. So we know how air gets into our body. I mean, we're pretty familiar, right? It comes in through our mouth and it comes in through our nose. When it comes through our nose, we say it comes through our nasal cavity. Now, it's the most, I don't want to say efficient, but the most beneficial way to get it through your nose. Why is that? Well, because we have mucosa in our nose, or snot in our nose, and the hairs, of course, which actually warms the air up before it comes into our lungs. Now, our nose are actually very vascular, hence why when you get a nosebleed, it kind of sucks. So we've got a lot of blood flow to that specific region, hence why our nose is predominantly very warm. Now, that warmth, that movement of blood flow in that specific area really helps to warm that air up, moisten that air before it gets into our lungs. Now, our mouth can do the same thing, but it gets dry very quickly and it loses that ability to make that air warm. And you may have noticed when you were running, back when you were a kid, long distance running out in the cold, and you actually get a dry, sore throat from just breathing in all that cold air. However, if you tried to breathe in through your nose, it wouldn't actually hurt as much because you've got that ability to warm that oxygen or that air up. However, the reason why we consume air through our mouth is obviously because we can get a lot more in a shorter period of time. So in that respect, obviously, it's important that you breathe through your mouth when you're running or when you're doing excessive aerobic exercise. However, it is obviously beneficial to breathe through your nose as well. Now, guys, I'm going to bring you to a little bit of a, because it's very clear here. If we look at the left bronchus, right, or the left lobe of the lung, and we call this the left side because obviously if it was sitting on you, this would be on your left side. So we do it in the plane of the individual, not the plane facing the individual. So if you look at the left bronchus, we can see this notch, this thing that's just like, it almost looks like it's cut out of the lung. Now, who can tell me what that is? Now, obviously, I'm not expecting an answer because no one's around me, but this is actually called, and you guessed it, the cardiac notch. Essentially, it's where the heart is situated. I remember updating my first aid course maybe about a year ago now, and the lady was telling me the heart is directly in the center of the chest. And she was a nurse, been a nurse like 40 years or something. She's an old bird, and I was sitting there like, what are you talking about? Everyone knows that the heart is slightly deviated to the left of the center of the chest. And she's like, no, it's directly in the center of the heart. I was like, okay, whatever. I'm not going to argue with her, but we know that it's actually direct. It's slightly deviated in the left side of the individual's body, hence why the cardiac notch is on the left lung and it's not on both of the lungs. But anyway. So we call this phase or this area where we breathe in and it comes into our lungs, into our bronchioles, into our alveoli. We call this the conducting zone. However, the respiratory zone is when it it diffuses from the alveolar sacs, the alveoli itself, into the capillaries, and vice versa, from the capillaries into the alveoli. So obviously we know that alveoli, when we breathe in, is housing specific gases, and the gas that we need is obviously oxygen. Now when it holds that gas, those capillaries that are wrapped around those specific alveolar sacs, actually that oxygen diffuses into those capillaries. However, the byproduct of the cellular debris and cellular waste is carbon dioxide, right? So that carbon dioxide is diffused from the blood vessels into the alveolar sacs so that we can exhale that carbon dioxide. So what does that essentially mean? Well, these blood vessels go through a phase of being deoxygenated to oxygenated, from, I suppose you can call it, blue blood to red blood. Okay. Now when it travels to the alveoli, it's containing deoxygenated blood, but when it travels back to the heart, it's containing oxygenated blood. Now that when it travels to the blood and containing the oxygenated blood, we know that blood is going to be pumped in the heart and then exerted or pumped out all throughout the body. So this is called the respiratory zone. But what makes capillaries so special? Well, they're actually a single cell layer membrane thin, so they're actually not very thick. That allows for the diffusion process to occur very rapidly. So bronchi, it's an interesting looking thing. I always look at a bronchi and I think it looks like a starfish. Sorry, not a starfish, sorry, a seahorse. But I could just be, imagining things. Anyway, so the bronchi is approximately 25 orders of branching, and the three main branches are the primary, the secondary, and tertiary bronchi. And the primary bronchi, which is the main bronchus, is the largest bronchus. The right side is wider and shorter than the left. The secondary bronchi is lobar, or referred to as lobar. There's 3 on the right and only 2 on the left. And you can start to think why this may be. And then we have the tertiary bronchi, and the bronchioles are less than 1 millimeter in diameter. And there's the terminal bronchioles, which are less than 0.5 millimeters in diameter. So you can start to think why the left may be smaller because of the cardiac notch, maybe. And then we have the terminal bronchioles, which branch into the respiratory bronchioles, which divide then into alveolar ducts. So what about the respiratory zone structures? Well, we have the respiratory bronchioles, and this is the branch from the terminal bronchioles, and it contains obviously those alveolar sacs, and it's the terminal end of the respiratory tree, and we say it kind of looks like grapes, sacs of grapes. So we have alveolar ducts, alveolar sacs, and alveoli. So alveoli are cup-shaped. They kind of look like grapes or little trees, except upside down. And it's the main site for gaseous exchange to occur. And this is the external respiration, i.e., between the lungs and the blood. Remember that the internal respiration, even though that we're diffusing the oxygen from outside of the atmosphere into our lungs and then into our blood itself, that's still considered external respiration. The internal respiration part is obviously when it diffuses into the cell itself. So remember, the capillaries are very thin membrane, and so are the alveoli. This allows for rapid diffusion of oxygen and carbon dioxide between the capillary blood and the alveolar airspace. This is cool. So it's lined with surfactant. This prevents alveolar collapse. So it keeps it hydrated, and it keeps its fluid levels up. And this allows for the lines of alveoli in the smallest bronchioles. So this image here is just a little bit of a detailed image regarding the cells, the endothelial cells, the endothelium, the alveoli wall. But essentially, this is the image here that you want to really be familiar with. So you can see that the alveoli, which is here, the alveolus, and this is the blood itself, the capillary, okay, is brushing past the alveoli. Now, essentially, because it's so thin, it allows for rapid diffusion to occur. Oxygen is transmitted from the alveoli into the blood, and carbon dioxide from the blood is transmitted into the alveoli. Hence, this diffusion process occurs. So the diaphragm, or diaphragm, obviously pronounced diaphragm, is a muscle that is used to allow the alveolar space, or the lung capacity, to increase and decrease with inhalation and exhalation. So we can see in the depiction here that we have an inhalation and the diaphragm contracts. So we have a relaxation and the diaphragm shortens and closes off the lungs. Okay, so this is the process of exhalation, inhalation, exhalation, inhalation. Okay. So this is the muscle separating the chest and the abdomen. Now, inspiration, the diaphragm contracts and increases the thoracic space. Air then enters the lung capacity. We then have expiration, which is when the diaphragm relaxes and decreases the thoracic space. Air flows out. Excuse me. Back now. So just a moment ago, we touched on diaphragmatic breathing in terms of how your diaphragm contracts and relaxes to allow your body to essentially inhale and exhale, or inspire and expire the external gases in the external environment. So we obviously have to understand that we're not only just sucking in oxygen when we inhale, we're also sucking in all the other gases that are in the atmosphere at that any given time. So we just went through all of this. So let's move on to the conducting versus the respiratory zone of the lungs. So just moments ago, I discussed the differences partly between the conducting and respiratory zone, but let's kind of just go back to that a little bit more specifically. So if you remember, the conducting zone provides fairly rigid conduits for air to reach the gaseous exchange sites. In this pathway, it also cleanses, humidifies, and warms the incoming air. So I may have mentioned earlier that when we inhale through our nose, we actually have all those nose hairs, the mucosa, the snot within our nose to actually help warm and cleanse that air as it comes into our lungs. I don't know if you've ever run a 400 meter sprint, or you've gone through a 1200 meter track or whatnot when you're doing exercise, and on a cold winter's day, and you'll notice that you'll get this dry sensation at the back of your throat, or almost sometimes into your lungs. That can be from a variety of factors, but one of the most common ones is because you're actually consuming your oxygen through your mouth. And when we consume it through our oral cavity, we don't have enough time to warm and humidify that incoming air into our lungs. So hence we get that dry, cold, sometimes painful feeling down in our lungs. That's not necessarily bad. It's just saying that you've got to try and warm that air up a little bit more. So that's just your body's way of saying stop breathing through your nose. However, the reason why we go from moving from breathing into our nose to breathing through our mouth is because we require more oxygen. So we know when we're doing exercises like that, our body requires oxygen to be able to perform the exercise. If we remember back to our metabolism lab and lecture, you would have remembered that when we go through aerobic glycolysis, or when oxygen is present with pyruvate can enter the mitochondria and creates X amount of ATP, around about 32 millimoles of ATP. So when we have those 32 units of ATP, are allowing us essentially to perform exercise. However, oxygen is required, so hence why we breathe through our mouth. Now, when we discuss the respiratory zone, the respiratory zone is the actual site of gaseous exchange. So this is essentially where the air is diffused between the alveolar sacs and the capillaries, the blood flow capillaries that surround or encapsulate those alveolar sacs. This is called external respiration. Um, so we have microscopic structures, and we have respiratory bronchioles, alveolar ducts, alveolar sacs, and alveoli, which allows us to perform gaseous exchange of the alveoli or the alveolus into the capillaries. So we get that exchangement of carbon dioxide from the capillaries into the alveoli and oxygen into the capillaries to create oxygenated blood, that travels back to the heart. So in this image here, we can see the conducting zone and the respiratory zone being at the alveolar sacs. Okay, so actually within the lung itself. Okay, so let's talk about ventilation. So ventilation happens in a series of two phases, inspiration and expiration, but essentially this is commonly referred to as breathing. Breathing is pulmonary ventilation. So pulmonary means related to the lungs, and we'll touch back on that when we look at the cardiovascular system. And it's actually a good thing to remember. So whenever you hear the term pulmonary, remember it's relating something back to the lungs. It's a good way to think about the heart, and we'll look at that a little bit later. So remember, there's two phases: inspiration and expiration. There's mechanical forces which cause the movement of air, which come into the lungs and outside of the lungs. And the mechanical forces are your diaphragm contracting and your intercostals and all the other muscles that are required to perform inhalation and exhalation. But you also have obviously a high pressure to low pressure gradient. So we know that gases, when you put them in the atmosphere, they always flow in the direction from high pressure to areas of low pressure. Hence why if you spray a deodorant can in the air, the deodorant just doesn't clump together, because that's where the high concentration is. They want to spread out. They want to diffuse across the atmosphere. So for air to enter the thorax, the pressure of the air in the thorax has to be lower than the atmospheric pressure. And we'll touch on that when we look at Boyle's law, which apparently we're doing right now. Okay, so Boyle's law essentially states that there's an inverse relationship between the volume and the pressure. So an inverse just means that when one goes up, the other goes down. So for movement of air into and out of the lungs. So as you can see here in step 1, as volume increases, pressure actually decreases, and vice versa, as volume decreases, pressure increases. So if you see this image here, it's actually quite a good image because it actually depicts it really, really well. So we can see here, these are like little ions or little atoms inside essentially a beaker, okay, and this beaker has a pressure gauge connected to it. So we have a piston, or we have some sort of plunger that pushes down. Okay, so at the moment the volume of the beaker is very, very large, right? So it's 1 liter apparently. Okay, and that gives you 1 atm of pressure. Okay, so not much pressure because it's in a large container. But as soon as we push down, we plunge down, we reduce the amount of volume or the size of that beaker to half a liter, we actually get an increase in the pressure. And that's because the atoms and the molecules inside the beaker is getting closer together. So now they're hitting against each other, they're being more aggressive, they're more violently interacting with each other, therefore or henceforth increasing the pressure. So I hope that kind of makes sense. And we can see this inverse relationship, or this inverse curve here, on pressure and volume. So spend a little time on that, guys, if you don't kind of understand it. But you can see that if pressure is 1 and volume is 1, then pressure is 2 and volume is 2. So essentially, as pressure increases volume. As pressure increases, volume decreases, and vice versa. As volume increases, pressure decreases. So let's talk a little bit about inspiration and expiration. Now, I know this is like a very, very easy concept to understand. We do it every single day. We don't have to think about it. It just happens. But for your level of knowledge, let's just kind of go through it. So inspiration, so these inspiratory muscles contract. They can be the diaphragm, could be a whole heap of intercostal muscles. Pec minor sometimes has an important role in it if you're very, very fatigued and so forth. But you can look at a little bit more of the inspiratory muscles. And in fact, we'll talk about that if I have you for human anatomy. There's also a thoracic cavity, so the volume increases, i.e. the lungs are stretched. Intrapulmonary pressure decreases. Air flows into the lungs from high pressure atmosphere to low pressure into the lungs. So this is kind of like the step-by-step process.
The opposite mechanisms actually occur for expiration. So you go from airflows into the lungs from high pressure, except now now they're going. It'll be expiring through because the pressure inside of our lungs is greater than the pressure outside of the atmosphere. So henceforth, the flow of air goes from inside the lungs outside into the atmosphere. You don't really know too much of pleural space and the pleura, but if you've ever heard of pleurisy, or anyone in the class have had pleurisy before, essentially, to my understanding, it's actually inflammation of the pleura surrounding the lungs. So have a look about that. It may be interesting for you to look up if you've ever had pleurisy in the past. I suppose you can kind of go through this guy step by step. I won't go through it myself, but you can just see the differences between from at rest to inhalation to exhalation, and the movement and the changes of the muscles as you do so. Okay, so we're going to be touching on lung volumes and capacities, and you should have definitely gone through this in a lot of detail in your lecture, so I'm not going to spend too much time on this one. But essentially, as you can see here, you've got residual volume, minimal volume, expiratory reserve volume, but essentially these are the fluctuations of inhalations and exhalations that we perform on a regular basis. So, amount of air inhaled or exhaled with each breath under resting conditions. Okay, so we have inspiration, expiration, and total lung capacity. So these are the three things you really need to be familiar with: IRV, ERV, and TLC. So inspiratory reserve volume is the amount of air that can be forcefully inhaled after a normal tidal volume inhalation. Expiratory reserve volume is the amount of air that can be forcefully exhaled after a normal tidal volume exhalation. And total lung capacity is the residual volume plus the tidal capacity. So in males, we can hold up to approximately 6 litres, and females approximately 4.2 litres. So we can just talk ourselves through that. But once again, you should have gone through a lot of this in your lecture slide. So let's talk about lung volumes and capacities for a moment. So remember, residual volume is the amount of air remaining after every forceful exhalation. Minute ventilation, or VE, is essentially tidal volume times the breath frequency. So for example, if you have 500 milliliters times by 12 beats per minute, 12 breaths per minute, we'd approximate 6 litres per minute. So anatomical dead space. So this is the volume of air inspired, but not involved in gaseous exchange in the alveoli. So this could be approximately 150 milliliters if the tidal volume is 500 milliliters. Alright, so who's heard of spirometry before? Probably most of you, if not all of you, have performed some sort of spirometry test, back when you were in primary school, high school, when you were very young. Usually most, pediatricians or doctors give you some sort of very basic spirometry test just to assess your lung capacity, your lung function. One of the old tests was you have this big pipe, put your lips around it, and you blow into it as hard as you can, and bubbles will essentially rise up. And obviously, the more bubbles you can rise up, the stronger your breath is, the more, the larger your tidal volume is, and all those type of things. So, pretty interesting, I suppose. Saying that, putting your lips around a big pipe and blowing bubbles up in the air isn't maybe the best analogy, but essentially that's what the test tests. So just maybe look up some spirometry tests just to wrap your head around if you have, or if you have seen one before. So it's the most common pulmonary function test, measuring lung function, specifically the amount of volume and speed of flow of air inhaled and exhaled. So with a spirometry, we can, I suppose, diagnose restrictive disorders, so reduced pulmonary compliance, and we can see that as an appearance as a reduced vital capacity. We also see obstructive disorders, so this is blockages or narrowing of the airways. We can obviously identify if it's more difficult to inhale or exhale given the amount of air. We can also measure by forced expiration volume in 1 second, so that's FEV1, so forced expiratory volume in 1 second. And then we can also determine the percentage of vital capacity that can be exhaled in 1 second, i.e. 75 to 85% in 1 second for healthy adults of obviously your vital capacity. So a flow volume and loop. So I'll see if we have an actual. Okay, so cool. So a flow volume and loop chart just looks something like this. So we can see a This is essentially what you're looking at is the speed at which your flow rate, or you can excessively or expire, into the tubing at one point. So we can see in normal we can expire a lot of gas straight away, and then we can maintain it quite on a nice ready descent. However, we can see with early small obstructions it changes slightly. We can see the changes as we go through. So these are something that you may need to be aware of, guys. Okay, so let's talk about Dalton's law of partial pressure. So this is a fairly easy concept to understand. So just listen up. So it's used to determine the individual pressure of each gas in a mixture of gases. So i.e. the total pressure of a mixture of gases is the sum of the individual partial pressures. So the partial pressure is equal to the individual pressure of 1, the individual pressure of 2, the individual pressure of 3 of those molecules, gases, or whatever it is. So the partial pressure is everything combined. So the pressure is exerted by each gas. Partial pressure of the gas is directly proportional to the percentage of that gas mixture. So let's have a look. Okay, so example, air. So as we know, air isn't just all oxygen, but we know the atmospheric air pressure is 760 millimeters of mercury. So air is comprised of nitrogen, oxygen, carbon dioxide, and water, and we can see that the atmospheric pressure is 760 millimeters because if we look at the partial pressure of nitrogen, the partial pressure of oxygen, water vapor, carbon dioxide, and the partial pressure of other gases, we get an accumulated partial pressure of 760 millimeters of mercury. So partial pressure at sea level is the atmospheric air. With atmospheric air, we can see nitrogen. We can look at all these here, guys, and I'm not going to talk them out to you. And we can also see this in alveolar air, so within our lungs. So it changes slightly, but it's very similar. Okay, guys, moving on. So just notice the oxygen content. Okay, Henry's law. So I have a video here. So guys, feel free to type that in on YouTube and watch it. But otherwise, I'm going to just talk you through it. But please, guys, if you don't understand Henry's law, I'm not going to go into too much detail. We obviously ran this through in the lecture. But yeah, if you want a little bit more information, if you didn't quite understand it, didn't grasp the concept, please watch the video. So Henry's law essentially states that when a mixture of gases is in contact with a liquid, each gas will dissolve in the liquid in proportion to its partial pressure. I.e., the greater the concentration of a partial gas, the more and the faster that gas will dissolve into the solution in the liquid. So it's also dependent on the solubility and temperature of the gas. So example, oxygen from alveoli dissolving into the blood. A few more examples. So the Henry's law is essentially the air-blood barrier. So the respiratory membrane is where gaseous exchange occurs, and this single cell layer membrane allows for easy transfusion or easy diffusion, is a better word, of the carbon dioxide and oxygen in and out of the capillaries and the alveolar sacs. So step 1, oxygen diffuses from air within the alveolus into the blood in the capillaries. Carbon dioxide is diffused from the blood in the capillaries into the air in the alveolus. So guys, by the way, if you didn't understand Henry's law, please go and watch the video. It makes a lot more sense. So guys, let's look at the lungs now. So we're going to start at the top, at the capillaries. In fact, let's start at the right atrium and ventricle. So just down here, we can see the blue section. Now guys, just so you know, blood isn't actually blue when it's deoxygenated. It's still red. Okay, it's still red. It's not blue, but we make it blue because it's less oxygenated. Okay, and what makes blood oxygenated is actually the heme, the iron content within the blood. You know, like squid and octopus, they have blue blood, and the reason they have blue blood is because their metal component within their blood, like we have heme, so we have iron. I think they have they have a different one. I think it's copper. Yeah, I think it's copper. And copper is their metal component within their blood that houses the oxygen, so it allows them to survive. Hence why their blood is blue. Guys, I'm not a biologist. A animal biologist or a marine biologist. These are just general facts that I'm pretty sure I know and they're correct. But by all means, don't hold me on my word. So please check your information, check my source before you believe me and start preaching it to the choir. So guys, so let's start the right atrium and right ventricle. So it's just over here, okay? So think about the heart, and I know some of you guys may have never seen a depiction of a heart, but that's fine. Just think of, we know that there's an oxygenated side and a deoxygenated side. So think about the deoxygenated blood being pumped out from the heart and traveling to the lungs, okay? Now this is called the pulmonary artery. Now the reason it's called the pulmonary artery is because it's traveling away. Arteries tend to travel away from the body. And pulmonary, remember when we hear the term pulmonary, we're referring back to the lungs. So it's the pulmonary artery that the heart contracts, the blood goes through the pulmonary artery. Remember, this is deoxygenated blood. As it travels to the lungs, it's obviously the capillaries branch out, so it goes from an artery to an arteriole to capillaries to arterioles and then capillaries. And then once it's wrapped around the alveolus, it diffuses the carbon dioxide within the blood into the alveolar sacs, and the alveolar sacs diffuse oxygen into the capillaries and makes that deoxygenated blood now oxygenated. Okay, so that occurs from here. So from the pulmonary artery, it goes to the capillaries, diffusion occurs, carbon dioxide, oxygen, and so forth. We now have oxygenated blood, and it travels back through the pulmonary vein. Veins travel to the heart, okay? This now oxygenated blood can then be pumped around the body. Now, as we go to the systemic arteries, the systemic vessels, the muscles, the tissues, every single cell of our body that requires oxygen, okay, those single cells, they're going to diffuse their carbon dioxide into the blood, and the blood itself, the capillaries housing the blood, is going to diffuse the oxygen from that oxygenated blood into those body cells. So the byproduct, obviously, of the cell manufacturing products and moving around and doing its cellular functions, they create a byproduct of carbon dioxide. So that needs to be expelled, and it's excreted via the systemic pathway. So those capillaries then take up that the carbon dioxide, turn that blood deoxygenated, it travels back to the heart, and this process repeats again and again. To make that a little bit simpler, we start deoxygenated. The blood travels to the lungs. It becomes oxygenated via the diffusion of the arterioles. The alveoli. From there, the oxygenated blood travels back to the heart. The heart pumps that oxygenated blood around the body. The systemic vessels and tissues deoxygenate that blood again, and the oxygen travels into the body cells. From there, it travels back via the systemic veins into the right atrium and right ventricle, and it goes through this process again and again and again. We call this phase external respiration. Remember, when we're inhaling and expiring the gases from the external environment into the blood. When it goes into our tissues, we call that internal respiration. So inspiration and expiration of oxygen, carbon dioxide into and out of the body cells themselves. Okay, and this is all powered by the process of diffusion. So guys, let's just have a quick look and see how many more slides we got. So we got 31, we've got 40 slides, okay? So we've got 9 more slides to go, so not too long, okay? So bear with me, try and pay attention as much as you possibly can. Remember guys, I try to give you as many videos as possible, so just watch those videos that are presented on the screens, they will help you. So transport of oxygen. So oxygen dissolves in plasma at approximately 1.5%, but it's mainly bound to hemoglobin in the blood at 98.5%. So we have oxyhemoglobin, which is essentially the hemoglobin binded to an oxygen molecule, and then we have deoxyhemoglobin, which is hemoglobin without the oxygen molecule. We also have, so one hemoglobin molecule carries 4 oxygen molecules. If I had a whiteboard, I would show you how I'd depict for you how the process occurs and how you can actually, how I draw the heme group to show the binded oxygen molecules, but I don't have that luxury for you guys. So if you just Google oxyhemoglobin, you'll be able to see that chemical structure. So we can see this deoxyhemoglobin plus an oxygen molecule goes through the lungs, and we create oxyhemoglobin plus hydrogen ion. Okay, so hydrogen ions are essentially an acidic component and a byproduct of the process of oxyhemoglobin, and vice versa. When we have this process, as I was going to say, it goes both ways. This oxyhemoglobin and hydrogen ions bind together to create deoxyhemoglobin and oxygen. This is where the diffusion occurs. Okay, so the dissociation curves, guys, you're going to have to watch this video, okay? It's very important, okay? If the video doesn't work, then you just type in oxygen hemoglobin dissociation curve and you'll get a lot of information regarding what you need to know for the saturation of hemoglobin. So we know that there's a partial pressure of oxygen, but there's only so much oxygen that can diffuse into the blood because there's only so much amount of heme content, okay? But essentially, the greater the heme concentration, the greater the oxygen concentration. But yeah, so look, you want to definitely have a watch of a video of that to try and wrap your head around this process. But essentially, this wraps it up for you. So 10 partial pressures of oxygen allows for 13.5% of saturation of hemoglobin, and we go through that same process. Now, guys, if you just look here, so factors that influence hemoglobin saturation depend on a few factors. So 1, temperature, 2, blood pH, and 3, the partial pressure of carbon dioxide. So an increase in temperature and acidity or a reduction in pH increases the unloading of oxygen from hemoglobin. So essentially it decreases hemoglobin's affinity for oxygen. And the Bohr effect occurs, so more oxygen is released into the tissues. So if you guys haven't heard of the Bohr effect, it's essentially very similar to the dissociation curve. We use like a rise over run formulation if you guys have done that from high school. And you can just see the differentiation between the effects of pH, the effects of temperature, and the effects of partial pressure of carbon dioxide on the the oxygen saturation of hemoglobin of oxygen. Okay, so please kind of try and wrap your head around that, guys. It's a little bit intimidating, I suppose, at first, but if you just follow the curve, follow the numbers, I'm sure you guys will be able to work it out. Once again, we spent a lot of time on this in the lecture, so I'm not going to spend too much time trying to explain this process again. But essentially, the Bohr curve shifts to the right when there's an increase in temperature, there's an increase in acidity, and there's a partial pressure on carbon dioxide change. So remember the transport of carbon dioxide, so it's dissolved in plasma, approximately 7 to 10%. It's chemically bound to hemoglobin, approximately 20 to 30%. So carbon dioxide plus hemoglobin creates hemoglobin carbon dioxide. We also have bicarbonate ions in plasma, so approximately 70%. This joins water and forms carbonic acid. It dissociates into bicarbonate acid and hydrogen ion, and the hydrogen ions bonds to deoxygenated hemoglobin. So we can see this process just down below. Okay, so a few more slides, guys. We're almost done. So this isn't a very, very important slide, but it just gives you an understanding. It kind of gives you just an idea about, what's kind of occurring. So talk yourself through it. So carbon dioxide diffuses into the bloodstream, and 7% remains in the plasma of the blood. Okay. So remember, red blood cells are essentially floating in the plasma, right? Yeah. So remember, when you have, if you ever drawn blood before and you put it in the centrifuge and you take it out and you look at the separation of the blood, I may even have an image of the separation of the blood in this PowerPoint slide. If I don't, I definitely have it in the cardiovascular system. So we'll talk about that next time. But essentially, we can see the differences in red blood cell content, white blood cell content, and platelet content in plasma. Okay, so if we look here, so 93% diffuses into the red blood cell, and then from that, 23 binds to hemoglobin, forming carboaminohemoglobin. Okay, so just quickly rushing over that, okay, so we talked about the 23% that binds to hemoglobin, forming carboaminohemoglobin. So we also know that 70% is converted to H2CO3 by carbonic anhydrase or anhydrase. That H2CO3 dissociates into hydrogen ions and HCO3, which is then expelled and moves out of the red blood cell. Those hydrogen ions, however, are removed by buffers, especially for hemoglobin. So respiratory rhythm. So this is a cool thing, okay? So this essentially discusses the rate and depth of breathing that adjust to maintain levels of like pH, carbon dioxide partial pressure, and oxygen partial pressure. So we'll change the rate and frequency of our breathing depending on pH changes, carbon dioxide partial pressure changes, and oxygen partial pressure changes. So we're always trying to maintain what we can see on the screen. we actually have the most potent stimulus for breathing changes because of pH. So, for example, if acidity, if we get acidic changes or alkaline changes, then we'll either speed up or reduce our breathing accordingly. Same thing for carbon dioxide and oxygen. So guys, in summary, because we're pretty much done now, I just want to run you through all the content that we kind of touched on today. So remember, we summarised the conducting zone versus the respiratory zone. So remember the conducting zone being where we breathe in through our nose and our mouth and warm, humidify and cleanse that air. The respiratory zone being the point in our alveolus where we're diffusing the oxygen and carbon dioxide from inside the alveolus into the capillaries to inside the capillary into the alveolus. So that is the difference between the conducting and respiratory zone. We also talked about pulmonary ventilation. Pulmonary ventilation being obviously ventilation within the lungs themselves, and obviously there's the other ventilation where it's within the body. So at the tissue level. Okay. So I also remember Boyle's law, guys. So Boyle's law is pressure versus volume. So remember, there's an inverse relationship. So when pressure increases, volume decreases. And vice versa, when volume increases, pressure decreases. So we also remember the lung volumes and capacity. So remember resting. So tidal volume, FEV1. So force ventilation, all those type of things. Really, really important. Try and remember all those little definitions I gave to you a little earlier. I want you to remember Dalton's law of partial pressures. Remember, that's the sum of all the partial pressures within the atmosphere, and that gives you your total partial pressure. Remember Henry's law, so this is talking about diffusion. And then also remember, try and wrap your head around the oxyhemoglobin dissociation curve, essentially, and obviously the Bohr shift as well. So remember, the Bohr shift changes depending on a few different variables: carbon dioxide content, oxygen content, temperature, pH level, those type of things. And then we also talked about oxygen versus carbon dioxide transport. So just remember that, and we talked about this a little bit. So, and we'll talk about this again next week. But oxygen and carbon dioxide transport. So remember when we breathe in all that oxygenated air. Those capillaries that are wrapped around the alveolus, that oxygen diffuses into into the capillaries. That capillaries draws that oxygenated blood to the heart. The heart then pumps that oxygenated blood to all the systemic vessels, all the systemic tissues, all the cells of the body that require oxygen. Those cells then give a byproduct of carbon dioxide. So the carbon dioxide is diffused into the capillaries and changing that blood from oxygenated to deoxygenated. That deoxygenated blood then travels back to the heart where it's pumped out towards the lungs through the pulmonary artery. And when we hear the term pulmonary, we're referring back to the lungs. So those capillaries, the pulmonary artery then go into arterioles, then into capillaries, which wrap around the alveolus, diffusing the carbon dioxide within the blood into the alveolar sacs, and then this process repeats, and continues. So guys, try to wrap your head around everything we've discussed today. So question time, I know it says in activity 6.1 do these instead of what the question says. However, chapters and the text changes from year to year depending on the textbook you have. So guys, just go and do your lab manual, answer all the questions in your lab manual, answer all the questions that you didn't actually understand within the tutorial lecture. It's very important that you do that. And then also go into your textbook that you have and go find the chapter about respiration or respiratory system. Go to the back of that chapter, you have questions. Within those questions, try and answer every single one of those questions. If you can't answer them, go and refer back to the text and try to answer every single one of those questions. They're not easy questions, but they're good questions, okay? Now guys, that's the end of today's presentation, but before we do so, I just want to give you a little bit of a heads up. So guys, a lot of you are doing more than one unit, right? You're doing 2, 3, 4, 5 units for those who are overloading. And that's completely great. That's awesome, in fact. I commemorate every single individual that does more than 4 units. It's absolutely awesome. But just remember, guys, if you're at uni and you've got 4 units, or 3 units, or 5 units, and your contact time at uni is 20 hours per week, just remember, for every hour you spend at uni, you should technically do 1 to 2 hours of work at home, right? Just so you completely understand and you can actually hold that content in your noggin. So let's go at the bare minimum. So if you do 1 hour per week, so remember your contact hours at university is 20 hours per week. You now have 20 hours extra to do at home. That's 40 hours of university work per week. That's a full-time job, let alone your other jobs that you do as well. And I'm sure you guys
either have chores around the house. You also have, I'm sure you have your own part-time jobs or full-time jobs, whatever you guys are doing. And then, of course, you have, your social outings, being social at university, at the ECU events, being social with your friends, being social with your family, spending time with your family, cooking all your food, meal prepping, exercising, going to the gym, playing sport, doing other co-curricular activities. It becomes a very, very hefty task to be doing all these units and doing all this this extra review and research. So my suggestion would be, it's not about the quantity of time that you spend studying. It's all about the quality. It's all about the efficiency of your work. So try and do exactly what I said. Okay, you've been to the lecture. You've been to the tutorials. That's absolutely awesome. You've been taking notes as you go. That's even better. You've been writing questions as you go. I didn't quite understand that. What is this? What is that? What is the conducting zone? Okay, answer those questions you've already written. Okay, for the lecture and the tutorial, answer them. Write them down in a notebook or write them in the computer. Okay. Once you've done that, go to your textbook. Okay. Find the respiratory system or whatever chapter corresponds to what we're learning in any of the tutorials, and try to answer those questions at the back there. Try to answer all of them. I think it's about 10 to 20, sometimes 30. Try to answer all of them. They're quite short answers, so try to answer them all. If you can't answer them, refer back to the text and try to answer all of them. Okay, this will assure that you are going to get the best chance of getting a high grade in your end of sem and mid sem exams. So guys, just remember that it's not about, that may take you literally 20 minutes. That's not an hour. That's not 2 hours. 20 minutes. If you can do that, that's awesome. Okay. My suggestion would be, a lot of tutors will say, "Oh, do it immediately after your lecture. Do it immediately after your tutorial, so it's fresh in your head." I disagree with that phrase, because it's fresh in your head, and then you forget it the next day, even though you did the homework after. So give yourself a couple of days. Give yourself a couple of days for that to sink into your head. Do the questions and try and answer the questions 3 or 4 days after. Give your brain the ability to comprehend the information that you learned the previous day or the previous week, and allow yourself to challenge your brain to recall that information days later. That's the challenge. That's what studying is all about. Being smart with how you study and being efficient with your studying. Okay. So guys, that's it for me today for the respiratory system. Next week we'll be back with the cardiovascular system. So I'm looking forward to running you guys through that. But until then, have a great week and I'll see you soon. Bye.